Welcome to the deep dive, where we take that huge pile of information you send our way, all the articles, the research, the clinical data, and we try to boil it all down into one pure, highly potent shot of knowledge. And today, we are wading into a topic that affects, an incredible number of people. We're talking nearly 500 million globally.
Yeah.
We're talking about type 2 diabetes, T2D.
And we should be clear from the top, we're not here to just rehash the historical standard of care. For decades, really, the conversation around TTD was all about, well, slow managed decline.
Right. Control your blood sugar, try to mitigate the complications as they come.
Exactly. Today, we're charting, I think, a really significant philosophical shift. We're looking at the science that's moving the needle from lifelong management to achievable, medically recognized remission.
That's the key word, isn't it? Remission.
Yeah.
For so long, a T2D diagnosis, it just felt like a life sentence,
Yeah.
a permanent thing.
It did.
But the science, especially over the last 10 years or so, is telling us something completely different. It's telling a new story about the disease's mechanism, its causes, and crucially, its reversibility. So our mission today is, I think, pretty clear. We're going to do a deep dive into the source material you sent, looking at foundational causes of T2D, the root pathology. Then we'll explore the proven strategies for actually achieving that reversal.
And then we get to the really exciting stuff.
Oh, yeah. We're going to look at the truly advanced, honestly mind-bending regenerative breakthroughs that are changing how we might treat this disease at a cellular, even a genetic level.
But before we jump in, I think to set the stage for this deep dive, we need to anchor ourselves with a bit of a reality check.
Okay.
The potential for remission is absolutely real, but the challenge is significant. We analyzed this major, really diverse cohort study from 6 different healthcare systems in the U.S. It gives us a great look at real-world outcomes.
Not just a perfect lab setting, so hit us with it. What's the data that defines this challenge?
In this huge group of patients, all living with TTD, the overall rate of remission was, well, it was alarmingly low.
Oh, low.
Just 2.9%.
Well, okay.
And that's remission defined as keeping your HbA1c below 6.5% for at least a year with no glucose-lowering medication at all. So that number, 2.9%, it really highlights the systemic difficulty we're up against.
2.9% is. That is a tough number to hear. It tells you that for the vast majority of people, the disease is just, it's already dug in, it's entrenched.
It is.
But okay, let's look inside that tiny success group. Were there common threads? What made those people different? What characteristics did they share?
And that is where the data gets truly illuminating. Because what we found was that 92.1%, so almost all of those remission episodes, were strongly associated with one of 3 key clinical characteristics.
Okay, so there's a pattern. Let's unpack that.
Yeah.
What are those 3 things that seem to predict success, and what do they tell us?
Okay, so the first was a lower baseline HbA1c. That's your 3-month average of blood glucose, specifically having it less than 7.0%.
So, better control to begin with.
Right. The second was a very short duration of diabetes. In most cases, this meant less than 1 year since the initial diagnosis.
Catching it early.
Super early. And the third was being on minimal medication. This meant the patient was either only on metformin or no glucose-lowering drugs at all right before they went into remission.
So if you put those 3 predictors together, early stage, relatively controlled blood sugar, and minimal medication, it paints a really powerful picture, doesn't it?
It does.
Like the body is giving us this narrow but very real window of opportunity. If you can catch the disease right at the beginning before all those chronic processes take hold, your chances of actually reversing it they skyrocket.
That's it, exactly. It completely reframes the urgency. It's not just about intervention. It's about aggressive, rapid diagnosis in primary care.
It confirms that T2D isn't static. It's a progressive disease that builds momentum over time.
Those initial few months, that first year, that's the critical period. If a patient is already, say, 5 years past their diagnosis, or their baseline HbA1c is up at 9 or 10%, the metabolic damage is just so much harder to undo.
So the new approach has to be all about early intensive intervention.
That's the hallmark of modern T2D care. Absolutely.
Yeah. Okay, so given that narrow early window for remission, let's pivot and look at why. Why does the body break down so quickly in the first place?
Because T2D isn't really about high sugar, is it? I mean, that's just the symptom you see at the end.
That's the downstream effect, yeah.
Fundamentally, it's a disease of energy imbalance, right? chronic oversupply of energy and fat accumulating in all the wrong places.
That is the single most critical shift in understanding, and it's encapsulated perfectly by what's known as the twin cycle hypothesis.
Okay.
This hypothesis, which was largely popularized by the work of Dr. Roy Taylor, it basically argues that T2D gets triggered when a person's genetic predisposition meets chronic overnutrition and physical inactivity.
The modern lifestyle, basically. Pretty much. And those elements lead to two key things. One, visceral obesity, that's the fat around your internal organs. And two, crucially, something called ectopic fat accumulation.
Ectopic fat. So ectopic just means in the wrong place, right? We're talking about fat being stored in places it was never designed to be.
Exactly. Places where it directly interferes with how an organ functions. And for T2D, we're talking specifically about fat infiltrating the liver and the pancreas.
So the problem isn't just having too much fat, it's where that fat ends up.
Precisely. Because that excess fat sitting inside your liver and your pancreas is what triggers the twin cycles that really drive the whole pathology of T2D.
All right, let's break those two cycles down. Let's start with the first one, the liver.
Okay, so the first cycle is hepatic insulin resistance. What happens is when all this excess fat, we're talking high concentrations of triglycerides and other lipid metabolites accumulates in the liver just stops responding correctly to the insulin that's circulating in your blood.
It becomes resistant.
It becomes resistant. The liver's job is to produce glucose overnight to fuel your body, then to stop producing it when insulin signals that you've eaten. But a fatty, resistant liver, it just keeps dumping huge amounts of glucose into the bloodstream even when it shouldn't.
So the liver is basically ignoring the stop signal from insulin. It's going rogue and just flooding the system with sugar.
That's a perfect way to put it. And that contributes massively to that high baseline blood sugar.
Okay, so that's cycle one. What's the second cycle? The one happening in the pancreas.
So the second cycle involves the chronic stress and eventually the progressive failure of the pancreatic beta cells.
And these are the little factories in the pancreas that actually make insulin.
They're the workers, yeah.
Yeah.
They produce, store, and secrete insulin. And they're working overtime trying to compensate for the resistant liver and muscles. They sense all this high glucose and all the fatty acids in the blood. And at first, they respond by just massively overproducing insulin. They're trying to shout over the noise of the resistance.
It sounds like that classic factory floor analogy, right? The workers are being pushed into triple overtime to meet a completely impossible demand.
It's a terrible burnout scenario. And this chronic exposure to high levels of both glucose and fatty acids, it's actually toxic to the cells. It has a name: glucolipotoxicity and lipotoxicity.
A toxic work environment, literally.
Literally. This environment induces severe stress on the beta cells. And while they compensate for a while, this constant excessive workload just leads to exhaustion, to oxidative stress, and finally to a steep decline in their ability to secrete enough insulin. And that's when you see the clinical onset of T2D.
I want to stick with the beta cells for a second because the sources make a really interesting distinction between the number of cells, their mass, and how well they're working their function. How should we think about that?
That's a critical point, and it helps us avoid oversimplifying it to just, the cells are dying. Now, some studies have shown a decrease in the physical beta cell mass. Some reports say up to a 60% reduction in late-stage T2D. But most researchers now agree that it's the beta cell function, their ability to sense glucose and then rapidly release the right amount of insulin, that's more critical to the disease, especially in those early stages.
So it's less about the total number of factories shutting down and more about the remaining factories being just unable to do their job properly because of all that internal stress.
That's it. Beta cells are normally incredibly resilient. They're built for compensation. They have all these adaptive mechanisms, but they can only take so much chronic abuse from that glucolipotoxicity and the inflammation that comes with it.
So T2D really manifests when their ability to compensate is finally just overwhelmed.
It's the tipping point. And that stress isn't just glucose and lipids. The whole microenvironment around the cells becomes toxic. You have these pro-inflammatory cytokines, chemical messengers released from that dysfunctional fat tissue, and they compound the problem by inducing severe oxidative stress, which leads directly to beta cell demise.
You mentioned dysfunctional fat and inflammation. The source material really calls this out, doesn't it? This idea of chronic inflammation from adipose tissue, that visceral fat as a foundational driver of insulin resistance. They call it adiposopathy.
Adiposopathy, yeah. It's the concept that visceral fat isn't just inert storage space. It's a metabolically active, highly inflammatory organ. When your fat cells become overloaded and dysfunctional, they trigger this state of chronic, low-grade systemic inflammation.
And they're pumping out those inflammatory messengers you mentioned, the cytokines.
Exactly. And those cytokines circulate throughout the entire body, and they directly interfere with insulin signaling in the liver and the muscle cells. It's a crucial link. Excess visceral fat leads to chronic inflammation, which then directly makes insulin resistance worse.
And what's so fascinating here, and you see this in the source material, is that the idea of targeting this inflammation isn't some brand new cutting-edge thought. There's a powerful historical echo.
There really is. I mean, if you go all the way back to 1876, you can find records of clinicians using salicylates and salicylate sodium to treat glycosuria.
Which is just excess sugar in the urine, right?
Mhm.
A classic sign of what we now call diabetes.
Right. And salicylates, of course, are the chemical family that aspirin comes from. They are fundamentally anti-inflammatory agents.
Yeah, so wait, long before you understood anything about beta cells or insulin resistance, doctors just observed that if you reduce inflammation, it somehow helps the body handle sugar better.
Precisely. It's an incredible piece of clinical observation that connects the 19th century to 21st century molecular biology. Today, we understand the mechanism. High-dose salicylates can inhibit a key inflammatory pathway called NFU, and that improves insulin sensitivity.
The modern drugs are obviously more sophisticated.
But the historical insight just confirms it. Targeting inflammation is a deep, established principle in diabetes care.
Okay, so the core takeaway from the pathology section is just crystal clear. T2D is caused by this excess ectopic fat that's overwhelming our metabolic systems.
That's the root cause.
And that realization leads us directly to, I think, the most exciting part of this deep dive, the actual definitive reversal strategies. Because here's where it gets really interesting. The ability to achieve and sustain remission is almost entirely dependent on successfully getting that fat out of the liver and pancreas. So, what does the proven roadmap for that look like?
Well, the foundational evidence, really the gold standard we all rely on now, comes from landmark studies like the Diabetes Remission Clinical Trial, or DIRECT.
Direct.
That trial was monumental. It was, because it demonstrated conclusively that substantial rapid weight loss can restore normal glucose control by literally clearing the excess fat out of those organs.
Let's nail down what substantial weight loss actually means in this context. It's not just about, fitting into your old jeans.
No, it's about hitting specific biological thresholds that trigger a real metabolic change. We can bring it down. Moderate weight loss, let's say losing 5% or more of your initial body weight, that's usually enough to start improving insulin sensitivity across the system.
Okay, so 5% is good.
It's good, but for true, durable clinical diabetes remission, the data is very clear. You're typically looking at losing 10 to 15% or even more of your initial body weight.
10 to 15%. That is aggressive. It's difficult for sure, but the payoff is remission. Did the DIRECT trial have specific data on that link?
Oh, absolutely. Clinically significant weight loss, which in their analysis was defined as losing more than 3% of body weight, was associated with 57% of all the remission episodes they identified.
And what about the people who didn't achieve remission?
That's the other side of the coin. Among that group, only 37% had managed to lose more than 3% of their weight.
That's a stark contrast.
It's a huge difference, and it really proves that weight loss is the primary lever of control here. If you lose enough weight, the fat content in your liver and pancreas drops, those twin cycles start to reverse, insulin sensitivity comes back, and the beta cells can actually recover their function.
The connection is just undeniable. But the source material flags the single biggest challenge to this whole strategy, doesn't it? Durability. It seems like getting into remission is only half the battle. Staying there is the really hard part.
That is the crucial and honestly the sobering point. Remission isn't a permanent cure. It's a metabolic state that requires lifelong behavioral maintenance.
What did the DIRECT trial show on that front?
In the DIRECT trial, about a third of the people who successfully achieved remission at the 1-year mark had relapsed by the 2-year mark.
Wow.
And here's the really actionable detail. That relapse overwhelmingly corresponded with weight regain, specifically gaining back more than 2 kilograms.
2 kilos. So that's, what, about 4.5 pounds?
Yep, About 4.5 pounds. That 2 kilogram threshold is almost like a biological tripwire. Regaining even that small amount of weight seems to be enough to push the system back into that state of ectopic fat accumulation and restart the whole vicious cycle.
So what does that mean for how we should approach this clinically?
It means we have to shift our focus. It can't just be about the initial weight loss phase. We have to dedicate significant resources to surveillance and maintenance. It means early intervention, counseling, maybe those VLCD boosts the moment a patient starts to creep up over that 2 kilo threshold.
It just underscores that for T2D reversal, weight maintenance is the ultimate therapeutic goal.
It is the therapy.
Okay, let's pivot back for a second to identifying the best candidates for success. Beyond the weight loss itself, how can clinicians spot the patients who are maybe metabolically or genetically predisposed to achieve remission?
As we touched on earlier, early diagnosis is king. It's everything. Remission is overwhelmingly concentrated in patients who are at a very early stage. Our data showed that nearly 57% of all remission episodes were in people with a diabetes duration of less than 1 year.
57%.
Yeah, that's nearly 3 times the rate compared to people who've had the disease for 5 years or more.
What about their medication status? I mean, if a patient is already on 2 or 3 different drugs, does that dramatically reduce their chances?
It strongly suggests the disease is more advanced, yes. We found that most patients who achieve remission were either on no medication at all, which was a small fraction, about 4.7%, or they were on a single glucose-lowering drug. That group accounted for 91.4% of the success stories.
It just reinforces that idea that your metabolic flexibility is still high when your medication needs are low.
It does, and having a lower baseline glycemia and HbA1c below 7.0%, that was another powerful predictor. It occurred in about 45% of remission episodes, which suggests less chronic damage from high glucose levels.
The message is just indisputable, isn't it? You have to strike hard and strike early before the body's compensatory mechanisms are totally exhausted.
Exactly.
Okay, so let's talk about the how, the specific diet components of that strike. You see so much noise in the media about a single perfect diabetic diet. What does the actual clinical consensus say?
The consensus is deeply pragmatic, which I think should be really reassuring for people. There is no single magical diabetic diet.
Thank goodness.
The primary non-negotiable driver of metabolic improvement is calorie reduction that leads to weight loss. Full stop. Whether you achieve that calorie reduction through a low-fat diet, a low-carb diet, or balanced meals is really secondary to creating the energy deficit you need to clear out that ectopic fat.
So consistency and just sticking to a calorie deficit is more important than being dogmatic about macronutrient ratios.
Absolutely.
But that said, different strategies do offer distinct benefits beyond just pure weight loss, right? Let's start with one of the most studied, the Mediterranean diet.
The Mediterranean diet has just immense therapeutic value. It has strong evidence not just for improving glycemic control, which, you'd expect from any diet that causes weight loss, but also for really robust cardiovascular benefits.
And that's critical since T2D patients are at such a high risk for heart disease.
It's hugely important. It improves lipid profiles, it can lower blood pressure, and it has these really strong anti-inflammatory effects, which loops back to what we were talking about earlier.
And didn't we see trial data suggesting it can actually help people avoid medication altogether?
We did. One randomized trial that was highlighted in the sources showed that following a Mediterranean diet actually delayed the need for anti-hyperglycemic medications in high-risk people compared to a traditional low-fat diet. That is a massive outcome.
Avoiding or delaying pharmaceuticals just by changing your dietary pattern.
It's powerful stuff.
Okay, what about the very popular and often very controversial low-carbohydrate diets? What role do they play?
Low-carb diets, and that includes ketogenic or very low-carb approaches, they can be incredibly effective, particularly in the short term. They typically produce a much quicker drop in blood glucose compared to a high-carb, low-fat diet with the same number of calories.
And that speed is important, right? For motivation.
For motivation, absolutely. Yeah. And critically, it allows clinicians to safely and quickly reduce or even remove certain diabetes medications, especially things like insulin or sulfonylureas, which carry a major risk of hypoglycemia or low blood sugar.
So for a patient who's really motivated to get into remission fast, this can be a powerful tool.
A very powerful tool, as long as they have appropriate medical monitoring.
And what about plant-based diets?
Vegetarian, vegan approaches also highly effective. They can improve insulin sensitivity, they promote weight loss. They tend to be very high in fiber, which is great for blunting those post-meal glucose spikes. And one of the key benefits there is just the structure of the diet itself. High fiber plant foods often have a lower caloric density, which just naturally helps with the overall calorie reduction you need for weight loss.
The flexibility is really reassuring.
Yeah.
I do want to touch on one specific intervention, though. Intermittent calorie restriction. There was a recent trial that showed some pretty remarkable remission rates.
Yeah, this is a great example of how a structured, intense intervention can really maximize the chance for remission. It was a 2022 randomized controlled trial looking at intermittent fasting. The intervention involved a very low 800 kilocalorie restriction diet on roughly 5 days a week for a period of 3 months.
It's so intense, but for a short period. What was the outcome after a full year?
The remission rate was just striking. 47% of the people in the intervention group achieved 1 year remission,
47%.
compared to only 3% in the control group. It just demonstrates that for the right patients, these structured, short-term, highly restrictive protocols can be a powerful catalyst to achieve that metabolic shift needed to clear the fat and restore pancreatic function.
But adherence is obviously key, and that kind of extreme calorie restriction has to be medically supervised.
Absolutely has to be.
Okay, so diet is one pillar. Let's turn to the other non-negotiable, movement. What is the role of exercise in T2D reversal, and does it have to be tied to weight loss to work?
No, which is the truly wonderful thing about exercise science in this context. Physical activity, especially structured training, enhances insulin sensitivity, often without relying on weight loss at all. The mechanism is independent of the calorie deficit.
How does that actually work at a cellular level?
So exercise, especially a combination of resistance training and endurance work, it improves mitochondrial function inside your muscle cells. And it increases what's called insulin-stimulated glucose disposal.
So it makes your muscles better sponges for soaking up glucose.
That's a perfect analogy. Even if you don't lose a single pound, your muscles become far more efficient at utilizing the sugar that's in your blood. And that lowers your baseline glucose levels and reduces the demand on those struggling beta cells in the pancreas.
So if a patient wants to optimize their glucose control through activity, what are the official guidelines we should be emphasizing?
The guidelines really advise a two-pronged approach. First, you want at least 150 minutes of moderate intensity aerobic exercise per week. So brisk walking, cycling, something that gets your heart rate up.
Spread out through the week.
Spread out over at least 3 days, making sure you don't have more than 2 sedentary days in a row. And second, resistance training, lifting weights, using resistance bands, body weight exercises. That should be done 2 to 3 times per week.
And combining them is best.
Combining them yields synergistic effects. It's consistently shown to be the most potent prescription for overall metabolic health.
And for day-to-day acute glucose control, there's even data suggesting that the timing of that exercise matters, especially around meals.
It does. A lot of acute studies demonstrate that postprandial exercise, so for example, going for a 10 or 15-minute walk immediately after you eat, provides better acute glucose control. It helps to attenuate or blunt those sharp glycemic spikes that happen after you eat carbohydrates.
So you're using muscle contraction right when the glucose is surging into your system.
Exactly. You reduce the immediate burden. And while those short post-meal bursts are really valuable for long-term benefits to overall insulin sensitivity, you're still looking at longer durations of exercise, typically 45 minutes or more.
Let's just circle back one more time to durability. The science of getting into remission is solid, but the practice of sustaining it is the real challenge. What does that maintenance phase look like?
It requires a strategic and a very proactive surveillance plan. It means lifelong weight maintenance and glucose monitoring. It means dedicated patient support, regular check-ins, and being ready to intervene the second that weight starts to creep up.
Back to that 2-kilogram limit.
That's the key. If a patient gains back, say, 1.5 kilos, the clinician might recommend a brief medically supervised VLCD boost, a return to that 800 kilocal diet for a week or two, just to reverse that small gain and prevent them from crossing the relapse threshold. Sustained success really relies on recognizing that remission is an active, ongoing state you have to maintain.
And one final critical clinical caution before we move on. If a patient is starting these intensive diets while they're on high-risk medications, like insulin, the risk of hypoglycemia is huge.
Oh, it's absolutely essential to address that. Clinicians must be prepared to reduce or even discontinue those high-risk medications immediately when an aggressive weight loss diet starts. Preventing a severe low blood sugar event is paramount. It's not only dangerous, but it can completely derail a patient's confidence in their commitment to the new plan. So, okay, we know that lifestyle interventions, primarily that aggressive weight loss, are the cornerstone of achieving remission. But for many people, whether it's because they have difficulty with the weight loss or their disease is just more advanced, medications are still essential. So what does this deep dive tell us about how pharmacological treatments are evolving?
Well, it feels like we're moving beyond just, managing the numbers.
Yeah.
We're now using drugs to actively modify the course of the disease and even to help patients achieve that critical weight loss needed for remission. Let's maybe start with the traditional targets and then move to the newer classes.
Sounds good. So the traditional foundation is all about modulating the insulin receptors, the IR. This is the primary signaling pathway that's responsible for glucose uptake in your muscle, your liver, your fat tissue. Metformin, exogenous insulin, they all work through or alongside this pathway.
But the real evolution, the game changer, is in the novel drug classes that target the gut and the kidneys. They're either mimicking our natural hormones or, in some cases, creating entirely new ways for the body to get rid of glucose.
Precisely. Let's talk first about the GLP-1 receptors, which are targeted by the GLP-1 receptor agonists, or GLP-1 RAs. These drugs are, well, they're revolutionary because they're pleiotropic. They do many different things at once.
Okay.
So they enhance insulin secretion, but they do it dynamically. That means they only boost insulin production when your blood sugar is high, which massively reduces the risk of hypoglycemia. They also slow down how quickly your stomach empties, which makes you feel fuller longer. And they seem to promote beta cell survival.
And this class has completely changed the conversation around T2D treatment, largely because of its effect on weight, right?
Absolutely. Many of the older T2D drugs actually caused weight gain, which was, counterproductive.
Terribly so.
GLP-1 RAs caused significant weight loss. And the high-dose versions, like semaglutide at 2.4 milligram, are now being incorporated specifically to help patients achieve that substantial 10 to 15% weight reduction we know is often needed for remission.
So they're not just glucose-lowering drugs anymore. They're powerful tools for metabolic modification, helping people stick to the energy deficit they need.
Exactly. And the other real game changer is the class of SGLT2 inhibitors, or SGLT2I.
Yeah.
Mechanistically, these are just completely different from anything we had before.
How do they work?
They're unique because they act solely on the kidneys. SGLT2 is a protein channel in the kidneys that reabsorbs glucose back into the bloodstream. SGLT2 inhibitors simply block that channel.
So the body just pees out the excess sugar.
That's essentially it, yeah. You excrete the excess sugar through your urine. In a sense, they're completely bypassing the whole insulin resistance problem.
But their importance goes way beyond just lowering blood sugar, doesn't it?
Yeah, correct. I mean, the most powerful data for those GLT2 inhibitors is in their demonstrated cardioprotective and renoprotective effects, their ability to protect the heart and the kidneys.
Which are the two organs most ravaged by long-term diabetes.
Exactly. T2D is the leading cause of end-stage renal disease and a massive risk factor for cardiovascular disease. These drugs dramatically reduce the risk of hospitalization for heart failure, and they slow the progression of chronic kidney disease. This class has fundamentally changed the calculus of T2D care. They're now foundational for patients with cardiovascular risk, sometimes regardless of their actual blood sugar status.
So moving from the drugs to the goals, our source material really emphasizes that those old standard HbA1c targets are giving way to much more individualized, personalized metrics.
Individualization is absolutely key. The general HbA1c goal, it's still under 7% for most non-pregnant adults. That's what you need to reduce the risk of microvascular complications like retinopathy and neuropathy.
But that target is flexible.
It has to be flexible based on the patient's age and their other health conditions, their comorbidities.
So for whom would we tighten that target? Make it more aggressive?
For a younger patient, someone with a long life expectancy and few existing health problems, the target should be tighter, ideally less than 6.5%. The idea is to maximize the amount of time they spend in a healthy metabolic state to reduce that long-term accumulation of damage.
And conversely, when do we relax the target?
For older patients, or for those with extensive existing comorbidities, the target is often relaxed to, say, 7.5 or even 8%. And the reason is simple: safety. You want to mitigate the risk of dangerous hypoglycemia.
A low blood sugar event in an older, frailer patient can be catastrophic.
It can lead to falls, fractures, terrible outcomes that far outweigh the minor long-term benefit of squeezing their A1C down another half a point.
We're also seeing technology, specifically continuous glucose monitors or CGMs, playing a much bigger role, even for patients who aren't on insulin.
CGMs are invaluable. They move us beyond that simple snapshot of an A1C. They provide real-time metrics like time in range or TIR.
TIR.
Yeah, TIR is the percentage of time a patient spends in their optimal glucose range, which is typically between 70 and 180 milligdl. A goal of having your TIR be greater than 70% is becoming a new standard of care.
What's the real advantage of using a CGM over just a traditional A1C test every 3 months?
The CGM gives you real-time feedback. It reveals glycemic variability, those dangerous high and low swings that an average like A1C can hide. And even using a CGM for a short time, say for just a few weeks, serves a powerful educational purpose.
Also,
It shows patients exactly how their individual food choices, their stress levels, their activity, how it all directly impacts their sugar levels in real time. And studies show that this educational piece alone can improve A1C by 0.4 to 0.7%, just because the patient becomes a much more active and informed participant in their own care.
that's incredible. Finally, let's just loop back to the first-line medication, metformin, and a critical safety check that was mentioned.
Yes, absolutely. Metformin is safe, it's effective, but it is known to interfere with the absorption of vitamin B12. So clinicians have to ensure they are monitoring for B12 deficiency annually.
And why is that so important?
Because the deficiency is insidious. Its symptoms, tingling, numbness, nerve damage, can perfectly mimic the symptoms of diabetic peripheral neuropathy. So, regular monitoring ensures we can catch it early, treat it with simple supplementation, and avoid misattributing a preventable medication side effect to the progression of the disease itself.
All right, this is where we shift gears and get into the truly experimental, the groundbreaking work that's happening in the lab right now. We're moving beyond just optimizing the treatments we have today and into the realm of things like genetic risk stratification and full-scale regenerative medicine. If the last section was about treating T2D now, this section is about how we might prevent and even cure it in the next decade.
This really represents the bleeding edge of precision medicine. And we can start with genetics, which is moving way beyond just asking about your family history. We're now talking about polygenic risk scores, or PRS.
Polygenic risk scores.
These scores use genome-wide information. They analyze thousands of different genetic markers to predict a person's risk for T2D with much, much higher accuracy than traditional methods.
How is that actually being applied in the clinical context right now?
Well, it's particularly useful for really high-risk groups. For instance, women who have a history of gestational diabetes, GDM, are at a dramatically increased lifetime risk for developing T2D.
Right.
Applying a PRS to this group can provide a highly personalized, quantifiable measure of that risk, and that allows for proactive, intensive lifestyle intervention and surveillance long before an actual diagnosis would ever occur. It's the essence of precision prevention.
So genetics aren't just predicting who gets the disease, they're also predicting who responds best to treatment, which is critical for that durability problem we talked about earlier.
Absolutely. The source material highlights some specific genes that appear to be key players in weight dynamics. We see genes like NEGR1 and BDNF, that's brain-derived neurotrophic factor, which seem to predict weight regain regardless of what intervention you use.
Wow. So some people are just genetically predisposed to have this metabolic pullback toward their previous higher weight.
It seems so.
That is a profound finding. If we can identify those people before they even start their remission journey, what does that change about the strategy?
It changes everything about the maintenance strategy. It doesn't mean weight loss is impossible for them, not at all, but it flags that patient as someone who's going to require highly intensive, targeted, and very likely pharmacological support for their long-term weight maintenance just to overcome that genetic pull toward relapse.
So you have to personalize the resource allocation.
You have to. We also saw that another variant, the PPRG variant, was associated with both greater short-term weight loss and a greater tendency for weight regain later. It just underlines the complex, sometimes contradictory role genetics plays in both success and subsequent failure.
Let's transition now to regenerative medicine. This is really fascinating, particularly how it's being used to tackle the most debilitating complications of T2D, like neuropathy and chronic wounds. The focus here is on stem cells, but not in the way most people think.
Right. This brings us to what's called the paracrine hypothesis.
Hmm.
The idea is that when you administer mesenchymal stem cells, or MSCs, most of their therapeutic benefit doesn't actually come from them turning into new tissue.
Okay.
Instead, they act like tiny mobile pharmaceutical factories. They secrete all these bioactive factors, including these little vesicles called exosomes.
And exosomes are like specialized cellular mail carriers,
Mhm.
right?
That's a great way to think of them. They are these tiny lipid-enclosed sacs, and they're filled with messenger molecules, microRNAs, proteins, growth factors. When they get released, other cells take them up and they can manipulate the local environment. They can reduce cell death, stimulate new blood vessel formation, and critically, they can dampen inflammation, which helps tissue repair.
And are they safe?
Well, so far, clinical trials involving systemic MSC infusion have generally demonstrated safety. No significant concerns yet about them forming ectopic tissue or tumors.
Okay, that addresses the major safety hurdle. So let's talk about that specific breakthrough with diabetic peripheral neuropathy using engineered exosomes. This study seems like a major step forward.
It's a remarkable proof of concept. What the researchers did was they engineered MSC-derived exosomes to be specifically loaded with a microRNA called miR-146A. They called these Exo-146A.
And they compared these engineered ones to regular naive exosomes.
Exactly. They used diabetic mouse models that were suffering from DPN, and they compared the two.
And what was the result of loading that specific microRNA into the delivery vehicle?
The engineered ones, the Exo-146A, showed substantially amplified therapeutic efficacy on neurological recovery. They actually shortened the required therapy duration compared to the naive exosomes.
How much shorter?
The mice treated with the engineered exosomes showed significant functional improvements, better nerve conduction, better response to stimuli after just 2 weeks. It took the naive exosomes 4 weeks to get comparable results.
That acceleration is the sign of highly optimized therapy. What was the mechanism? How did they do it?
The miR-146A loaded exosomes significantly suppressed chronic inflammation in the mice by inhibiting the toll-like receptor, TLR4 and FU signaling pathway.
The same inflammatory pathway we talked about earlier.
The very same. It's a central command center for inflammation. By suppressing it, the exosomes reduced that chronic damaging immune response in the nerve tissue, which allowed the neurological healing to finally proceed unimpeded.
And this is the critical, almost revolutionary finding from that study. The exosomes did not affect the blood levels of glucose or lipids in the mice. Yeah. Why is that distinction so important?
It means they are surgically precise. They are specifically targeting the neurological inflammation that is caused by the diabetes, not the underlying metabolic disease itself.
So for a patient who has good glucose control, but still suffers from agonizing neuropathy,
A very common scenario.
we currently only have symptomatic treatments. This engineered exosome could represent a targeted regenerative treatment for that complication that is completely independent of their day-to-day blood sugar. There's a total paradigm shift.
It is.
And we're seeing this exosome work being applied to other things too, like chronic diabetic wounds.
Absolutely. Exosomes derived from all sorts of cells, epidermal stem cells, immune cells, are showing real promise in diabetic wound models. They promote anti-inflammation, tissue repair, new blood vessel growth. And this is where another field, nanomedicine, also steps in.
How do nanoparticles help this process?
Well, nanoparticles and other nanomaterials, like liposomes, can be engineered for highly targeted delivery. You can encapsulate a potent anti-inflammatory agent inside one and deliver it directly to the injury site, bypassing the rest of the body and concentrating the therapeutic effect right where you need it. It helps create an optimal healing environment where natural processes often fail.
This level of engineering it's just incredible. It
Yeah.
moves us into advanced tissue engineering. The sources mention things like 3D bioprinting as a potential solution to the organ shortage problem.
The global need for donor organs just vastly outstrips the supply. 3D bioprinting aims to solve this by literally constructing functional, lab-grown tissues and organs. The key hurdle, though, has always been vascularization.
Getting blood vessels to grow through the printed tissue.
Exactly, to supply oxygen and nutrients. A tissue can't grow larger than a millimeter or two without an internal, functioning vascular system.
And there's been a recent success on that front.
A huge success. Researchers have successfully created human lung tissue organoids that can produce their own organ-specific blood vessels. This is monumental. It suggests we're getting closer to a future where we can use lab-grown tissues for direct repair or even replacement of damaged organs.
And finally, let's talk about the sharpest tool in the genetic shed, CRISPR gene editing. We now have confirmation that personalized genetic medicine is accelerating into the clinic, and fast.
This is perhaps the most profound leap of all. We analyzed the confirmed success case of infant KJ from 2025. KJ had this rare, life-threatening metabolic disorder called CPS1 deficiency.
What made this case so critical?
CPS1 deficiency causes a fatal buildup of ammonia in the blood. Typically, these patients need a liver transplant to survive, but they have to be stable enough, old enough. The waiting period is incredibly high risk.
And KJ was treated with CRISPR.
KJ was successfully treated with a customized, personalized CRISPR gene editing therapy. It was delivered via a viral vector, and it edited the genetic defect in the infant's own liver cells. It's what's called a somatic cell editing approach.
The significance here is twofold, right? Feasibility and speed.
Exactly. It confirms the technical feasibility of creating bespoke genetic medicines, and it dramatically accelerates the timeline for when we can deploy them. It shows we can potentially correct life-threatening genetic defects in patients who are too young or too fragile for conventional surgery. This is the foundation for correcting other metabolic disorders, including those that might predispose people to T2D.
But with this immense power comes immense caution. The sources rightly emphasize the FDA guidance on long-term follow-up.
Caution is paramount. We are talking about permanently modifying the human genome. The FDA guidance is very clear. Any gene therapy product that uses genome editing technology like CRISPR or integrating vectors like lentivirus requires long-term follow-up observations for up to 15 years.
15 years. What are the specific risks that require that kind of observation?
The primary risk, the genotoxic risk, relates to the potential for inducing DNA double-strand breaks in the genome, which could theoretically lead to cancer. There's also the risk of off-target mutations, or that the viral vector might insert itself in a harmful location in the genome.
And these problems might not show up for years.
Exactly. Since these adverse events can be very delayed, that long-term follow-up is crucial for safety. This technology is incredibly powerful, but we have to proceed with a deep sense of scientific humility about the rare long-term consequences.
This has been a massive deep dive. I mean, we've covered the entire spectrum of type 2 diabetes, from the cellular mechanism of failure right up to personalized gene editing.
The synthesis of the source material really does provide some key, actionable insights. First, that T2D is fundamentally a disease of ectopic fat accumulation, and that's what explains why it is definitively reversible in its early stages through aggressive, sustained weight loss.
Right. Ideally, 10 to 15% of your body weight.
Second, the roadmap for that reversal is now incredibly detailed. We know success is most likely early on, and that calorie restriction is the main lever.
But we also know that specific diets, like the Mediterranean, offer those distinct cardiovascular benefits, while low-carb diets can give you that rapid glucose reduction.
And exercise provides those unique weight loss-independent gains in insulin sensitivity.
And finally, looking ahead, treatment is becoming hyper-individualized. We're leveraging new drug classes, like the GLP-1s and SGLT2Is, that don't just control sugar, but actively modify the disease course. They promote weight loss, protect the heart and kidneys.
And the science is just moving at warp speed. We're talking engineered exosomes to treat neuropathy without affecting blood sugar, and precision gene editing to solve deadly metabolic disorders in infants. It's incredible.
It really is.
So we know remission is possible. It's achievable through disciplined lifestyle change, supported by modern pharmacology and technology. But that leaves one massive, urgent question hanging in the air for you to consider based on everything we've analyzed today. We have very strong evidence, especially from trials like DIRECT, that aggressive lifestyle changes can induce T2D remission, the normalization of blood sugar. The critical question that future research has to answer is this: can achieving and sustaining that metabolic remission permanently reduce severe macrovascular morbidity?
Things like heart attacks and strokes.
Heart attacks, strokes, kidney failure over the long term. And can it do so beyond the reduction we already get from traditional aggressive risk management using medications like statins and blood pressure drugs?
That is the ultimate test of this entire reversal science, isn't it? Proving that restoring your metabolic health gives you a dramatically longer, healthier life. Not just better lab numbers today. That's a powerful thought to leave you with.